All right, well, it's 1 o'clock. Teresa, what do you think? Is there anything that you'd like to say before we get started, or do you think we should wait a few more minutes?
I think we're good to go, and you can just go ahead and introduce and
All right, well, it looks like the waiting room is calming down a little bit, so why don't we go ahead and get started? I am here to introduce Chloe Hanson. I'm very excited and proud to do this today as she defends her master's thesis. And I'd just like to take a couple of minutes to say a few words about Chloe and the work that she has done in our lab. So Chloe has had what some might call a non-traditional academic path. After graduating college with a BS from OSU in 2012, Chloe took several years away from higher education to pursue organic farming. And she owned and managed her own business, a farm near Salem, for several years. And as she puts it, she spent a lot of time on tractors, listening to science podcasts, and became just compelled to come back to the university to earn a graduate degree in biology. So she sold her business and came back to OSU. So Chloe started working in my lab, in the Burke lab, in the fall of 2019. And in the Burke Lab, our projects are generally thematically connected in that we use the same experimental yeast system to investigate the genetic basis of various traits. And at the time that Chloe approached me to talk about projects, I had just read this really interesting paper about how the herbicide Roundup affects yeast. And I suggested this to her as a trait we might pursue. And at the time I thought, oh, this is a great idea. You know, our interests are perfectly aligned, me as a geneticist and her as a former professional farmer. But I noticed her face fall a little bit, and I realized that she was just frankly over farming and instead just really wanted to learn some hardcore genetics. But ultimately, we did decide that this was the right project for her to pursue. So today she'll be telling you about the work she has done, which sheds some light about the genetic basis of resistance to Roundup, and it does indeed involve some sophisticated genetics. And before I hand it over to her, I just have to say that Chloe has been a tireless warrior in the pursuit of her thesis. I have been just awestruck to observe her do the experiments for her project with meticulous attention to detail, despite the many, many challenges that COVID brought our way. She stayed on track, and she's put together what I think is a really impressive body of work in just over 2 years, through the shutdowns, through the essential research approvals, the masking and distancing in the lab, the advisor who was always at home supervising a toddler. She had to do 3 or 4 moves, sometimes across state lines. And through it all, Chloe has been professional, curious, rigorous, and just generally unsinkable. So I'm very sad that she's leaving the lab, but I'm also so excited to see what she does next. And I'm happy to report that she's been accepted to a PhD program in the Human Genetics Department at the David Geffen School of Medicine at UCLA, and she will begin her studies there next month. So without further ado, I'm going to let Chloe take over and tell you about the research that she has been working so hard on.
Well, thank you, Molly, for the introduction. I really appreciate you and the full Burke Lab. I've had a really great time here at OSU. And despite my burnout on farming, I am excited to talk to you about my research here, working with Roundup. But for those of you that don't know me, my name's Chloe, and I really thank you for coming today to hear about my research. So I'll be talking with you today about my research that was investigating the experimental evolution of Roundup resistance now crossing yeast. So a little bit of a background. Glyphosate, the main active ingredient in Roundup, is one of the most widely used chemicals in the world. Since the advent of GMO corn and soy in 1992, we've seen a drastic increase in glyphosate use. This plot shown on the left from the USGS shows corn shown in yellow and soy shown in green, that accounts for the spike in glyphosate use over the past 25 years. The map on the right spatially demonstrates Roundup use in 1992, and then again in 2017, over the course of which we've seen over a 5-fold increase. For my thesis research, I'm investigating the genetics of Roundup resistance in non-target eukaryotic organisms. The use of glyphosate is increasing worldwide, and the impacts of its application are currently understudied. I am interested in understanding the biological effects of its long-term use. There are ecological concerns over its use, especially regarding the effects of aquatic species with runoff from agricultural fields. Despite the manufacturer's claims of glyphosate safety, there are also concerns over human health, with recent legal battles linking its use to non-Hodgkin's lymphoma. Just last month, Bayer actually announced that it will stop the sale of glyphosate-based products to residential customers, including Roundup, due to this growing legal pressure. There are also agricultural impacts, both on food safety and the effect of soil microbial communities, which support plant health and productivity. So my thesis work aims at understanding how the model organism Saccharomyces cerevisiae evolves in the presence of Roundup. There is a need for more studies of the effects of Roundup on organisms other than plants to gain a better understanding of the general consequences of its use. Experimental evolution uses long-term selection experiments and full genome sequencing to investigate how organisms evolve under different conditions in the lab. For example, microorganisms can experience hundreds of generations of selection in a matter of weeks, where we can track phenotypic and genotypic changes over time. By comparing the genome and phenotypes that change of the evolved populations with respect to the ancestor and control populations, we can observe evolutionary dynamics in real time. S. cerevisiae is an ideal candidate for studying the effects of Roundup, as it is a non-target species that can be found in natural environments where herbicides are applied. And it is also a powerful model system for understanding how more complex organisms might be affected. It has a small genome size that can be cheaply sequenced and has abundant genetic variation within natural yeast populations. It has short generation time and is easily handled under laboratory conditions, making it feasible to test large pooled populations with millions of individuals. Populations also can be frozen in time and rejuvenated, which makes them an ideal candidate for studying evolution and genetics. And also, humans and yeast are separated by about 1 million years of evolution, but nonetheless share thousands of orthologous genes, making these a powerful model organism for investigating the health impacts of chemicals. And one of the aspects that is most interesting to my experimental design is that yeast can reproduce both asexually and sexually. They can bud off, creating a genetic clone of themselves, or they can reproduce sexually, undergoing meiosis and recombination. Evolution experiments that use isogenic haploid strains undergoing only asexual reproduction make it easy to track the accumulation of potential de novo mutations over many generations, as shown by the red marks that appear on a single chromosome after several generations of selection. Alternatively, in the Burke lab, we also are interested in modeling how standing genetic variation changes over time in more complex eukaryotic organisms. This is visualized in the bottom portion of the figure by the changing orientation of the different colors on the 2 homologous chromosomes over time. Each color represents standing genetic variation inherited from the different founders of the starting recombinant ancestral population. We can use highly diverse sexually reproducing diploid yeast populations to track both putative de novo mutations and the evolution of standing variants in the lab. In sexually reproducing eukaryotic organisms, adaptation is generally driven by the evolution of standing genetic variation and not beneficial de novo mutations. Therefore, the focus of this talk will be on how this genetic diversity evolves over time under Roundup selection. Starting with an experimental population that has a complex genetic history and continuing to induce outcrossing over the course of a selection experiment, leverages recombination's ability to break up linked hitchhikers, whether they be neutral or deleterious. This rapid decoupling helps to promote faster adaptation, allowing us to observe large phenotypic and genomic changes in a matter of weeks. The recombinant 12X population, whose founders are shown here in this unscaled phylogeny, was used in my master's research to test the effects of Roundup on yeast. It was developed by crossing 12 isogenic strains derived from unique environments and geographical locations that likely had different levels of chemical exposure before isolation. The colors highlighting the names of each of these founders represent their isolate locations, and the colors on the tips of the trees shows the different environments from which these isolates were derived. Using highly diverse populations such as this allows us to model the adaptive dynamics of standing genetic variation. The 12X ancestor was developed by crossing these 6 pairs. The resulting mated diploids were then sporulated, and then the spores were pooled equally among the 6 crosses for another 12 rounds of outcrossing. The resulting population has a rich history that may help us better understand the impacts of Roundup on more genetically diverse, naturally eukaryotic populations. Using these experimental protocols, my thesis work aimed to investigate 2 main questions. First, what genes are involved in Roundup resistance? I explored this question by monitoring changes in standing variation and identifying potential de novo mutations that arose over the course of selection. In plants, glyphosate targets the EPSPS gene of the shikimate pathway, which is a necessary part of amino acid production. It is well established that yeast and many other microorganisms also share the same pathway. However, it is not present in any known animal species. In yeast, it has been shown that different variants within the yeast AR01 gene, which is homologous to the EPSPS gene in plants, cannot fully explain the differential growth of yeast in the presence of glyphosate-based herbicides. This highlights the need for more research into its effect on eukaryotic organisms from a health perspective, since it appears that other physiological pathways are likely affected by Roundup exposure that may also be orthologous in humans. Further work performing traditional QTL mapping in yeast has also identified 2 candidate genes, PDR5 and ABC multiple drug transporter, shown on this peak on chromosome 15, and DIP5, an amino acid permease that was identified on chromosome 16. After further functional genomics work, it was hypothesized that these 2 genes might determine how Roundup gains entry into the cell. More recently published work has also identified more potential candidate genes through RNA-seq and mutation accumulation experiments in single isogenic strains. Over 1,000 genes were up or downregulated after exposure to glyphosate-based herbicides. And in the selection experiments using individual isogenic strains, 148 genes accumulated nonsense mutations by the end of selection. We were curious to see if we would detect similar signals at these loci and hope to also identify other potentially causative sites that contribute to Roundup resistance. The second question that we wanted to address is, will implementing an incrementally increasing selection regime enhance our ability to identify causative genes? This question was inspired by Blachus and Koffler's work using computer simulations to model the effects of different selection regimes during evolution experiments. This plot shows that a selection regime that incrementally increases, shown here in black, optimizes the true positive rate while decreasing the false positive rate, shown on the x-axis, compared to a range of different constant selection pressures, shown in colors below. To empirically test this hypothesis, we implemented 2 different Roundup treatments over a 10-week selection experiment using the recombinant 12X population with 16 biological replicates for treatment. The first was an increasing regime that started at 0.1% Roundup and increased by 0.1% each week until reaching 1% by the end of the experiment at week 10. Preliminary work demonstrated that our 12X ancestral population had a linear response to long-term Roundup exposure. Therefore, we determined that starting at a low dose of 0.1% would maintain approximately 90% of the genetic diversity in the population at the beginning of the experiment and would gradually decrease the number of individuals being selected for over time as the strength of selection increased. The second round of treatment was a constant high selection pressure of 1% Roundup in YPD media that we estimated would select for a 90% reduction in viability each week. The control with no Roundup exposure was also implemented in order to identify and correct for genomic changes due to routine yeast handling procedures. These three treatments all underwent the same weekly handling protocols and sexual outcrossing each week, except the control that was not exposed to Roundup. Roundup weed and grass killer 3, which contains 2% glyphosate, was applied weekly for 48 hours to both of the Roundup treatments. It should also be noted that our control populations were used for multiple radiative experiments in the lab, so today's genomic results will focus on comparisons between week 10 in the 2 Roundup treatments and week 15 in the control. The novel outcrossing techniques that we implement in the Berk lab allow us to model the evolutionary dynamics of populations with higher genetic variation compared to classic QTL and asexual selection experiments. The following diagram shows the weekly protocols that we use for selection and outcrossing. The 12X population was also developed using these methods, and they were used in our weekly protocols throughout our Roundup selection experiment. Step 1 involves 48 hours of selection in batch culture, where the yeast cells grew vegetatively and the Roundup treatment was applied. Cell concentrations were standardized at all transfer points to control for density effects on population growth and to ensure that population size of at least 1 million were maintained in each replicate to prevent drift. During step 2, sporulation was induced in minimal nutrient media, and in step 3, spores were isolated using mechanical and chemical agitation. The final step included letting the isolated spores randomly mate, and then they underwent a recovery period where several generations of asexual reproduction occurred. This weekly protocol was repeated for 10 consecutive weeks, and we estimate that over 120 generations of competitive selection occurred in both the increasing and constant selection regimes over the 10-week period. After 10 weeks of selection, evolved Roundup treatments in the control populations were phenotyped alongside the ancestor and the controls in Roundup, 1% Roundup media, and also in plain YPD media. Doubling time was assessed using Wilcoxon rank sum tests. Growth was tested in YPD media only without Roundup to see if there were any phenotypic trade-offs that resulted from Roundup selection. Full DNA sequencing was performed using Illumina's Nextera library preparation kits and HiSeq 3000, DNA paired-end sequencing of 150 base pair reads, and we strove for a greater than 50x average genome-wide coverage. Genomic data was analyzed by first aligning reads to a reference genome and calling variants using GATK version 4.0. SNPs then went through additional quality checks, and then a principal component analysis was performed on the allele frequencies of all SNPs with coverage greater than 10 that were common to both the evolved populations and the ancestor at each time point. CMH tests followed to further assess allele frequency differences between the two Roundup treatment groups and the controls. Over 55,000 polymorphic high coverage SNPs were used to perform each CMH test. 3 pairwise CMH tests were run. The first compared the final week's allele frequencies of the constant Roundup treatment to the control populations. The second CMH test similarly compared the final week's allele frequencies of both the increasing Roundup treatment and the control populations. These 2 tests helped to identify allele frequency changes that were likely due to Roundup selection, not routine yeast handling procedures. A third CMH test was also performed, comparing the constant and the increasing treatments directly to further verify if there were any allele frequency changes that were independent to just one Roundup treatment group. So now I'll talk to you a little bit about the results of my study. Our phenotyping results comparing the constant, increasing, and control populations to the ancestor after 10 weeks of selection show that all evolved populations, including the control, have improved growth in the presence of Roundup compared to the ancestor when doubling time was assessed. The plot on the left shows the growth curves generated by plotting absorbance measurements on the y-axis over time on the x-axis. The plot on the right shows the average doubling time of all replicate populations in each treatment group and the common ancestor population, estimated from these absorbance readings. The p-values from each pairwise comparison to the ancestor are shown on the box plot. The ancestor is in turquoise, the constant in red, the control in blue, and the increasing in green on the far right. Comparisons that significantly differed are star. The evolved Roundup treatment populations also grew faster than the control populations in Roundup. However, the 2 Roundup treatments grew at similar rates. We were surprised to see substantially improved growth in the presence of Roundup of the control populations compared to the ancestor. A specific aspect of our protocols that may have been playing a role in this phenotypic outcome is the use of Wiper, a detergent used to kill unsporulated diploid yeast cells during the weekly sexual outcrossing protocol. This chemical is of specific interest since it might be similar to undisclosed surfactants that are added to the commercial Roundup formulas in order to increase the coverage, penetration, and overall effectiveness of the herbicide. The acute Wiper exposure may be targeting similar physiological pathways as the non-active ingredients in the commercial Roundup formula during our weekly selection protocols. When we tested the evolved population's growth after 10 weeks of selection in YPD media without Roundup, we observed faster growth in the evolved control populations compared to both the ancestor and the 2 Roundup treatments. The evolved Roundup treatments also grew at rates comparable to the ancestor. And the constant Roundup treatment grew faster than the increasing treatment. The faster growth in the control populations compared to the populations treated with Roundup media may suggest a slight phenotypic trade-off due to Roundup selection, since both Roundup treatments appear to not grow as efficiently in nutrient-rich YPD media. However, no major trade-offs were observed since the evolved Roundup populations performed as well as the ancestor. When we look at the genomic data after performing a principal component analysis of the polymorphic SNPs, we see that all treatment groups are evolving from the ancestor. We also see that the constant increasing treatments have independent genomic signatures that change over time at weeks 3, 6, and 10. And finally, that the yeast have evolved under routine sexual outcrossing protocols as genomic changes were observed in the control populations. You can also see that there is not distinct clustering of individual replicates in the control as there is in both the constant and increasing Roundup treatments. This potentially could implicate the effects of genetic drift rather than selection. Further analysis of the allele frequencies after the final week of selection showed regions of the genome that significantly diverged in the constant Roundup populations from the control populations after performing a pairwise CMH test. The chromosome number and megabase position are noted on the x-axis of this plot, and the negative log 10 p-value is plotted on the y-axis. The higher negative log 10 p-values correspond to a greater divergence in allele frequencies between the constant and control treatments. The blue line represents a significant threshold that corresponds to a 95% confidence level that was derived using permutation methods, and peaks were defined by the significant SNPs above the prescribed cutoff. The second pairwise test comparing the increasing Roundup treatment to the control identified several chromosomal regions that also strongly deviated over time. And finally, a third test comparing the two Roundup treatments was performed to see where the constant and increasing population allele frequencies also diverged. We identified 4 strong candidate regions where there are peaks that are shared in both the constant and increasing evolved populations comparison to the control. We classified these as Roundup-specific peaks since these genomic signatures were common to both Roundup treatments despite the differing levels of Roundup exposure and did not significantly differ in our third CMH test comparing 2 Roundup treatments. There are 58 genes falling under these peaks, warranting further investigation of whether these genes may be contributing to evolved Roundup resistance in our 2 treatment groups. When we zoom in on the first Roundup-specific peak on chromosome 4 in the constant treatment, we see multiple significant SNPs above the threshold. The plot on the right shows the allele frequency change of the most significant SNP over time in all biological replicates, starting from the common ancestor frequency. The allele trajectories of each replicate within the constant, shown in red, the increasing, shown in green, and the control, shown in blue, visualize a clear divergence of the control from both the constant and increasing treatments, making this a strong Roundup-specific region. We interpret these results as evidence that these particular alleles or those linked to them may be under stabilizing selection in the 2 Roundup treatments, as they may remain at a high frequency throughout the experiment. The allele frequency trajectories in these peaks also show that the control population might be under weak directional selection as it diverges from the 2 treatments. The allele frequency trajectories in this peak are fairly representative of the majority of the SNPs trajectories in the other Roundup-specific peaks. However, more variation was found among individual replicates within the other peak regions. We also identified 2 constant treatment-specific peaks that were not identified in the increasing regime and were also found to be significantly different when the 2 Roundup treatment groups were directly compared, as demonstrated in the bottom plot. These 2 peaks highlight an additional 44 genes that might specifically relate to improved resistance to high Roundup exposure. There were no peaks that could be classified as increasing specific. When we take a closer look at the constant specific chromosome 4 peak, we see a handful of significant SNPs above the significance cutoff. The allele frequency of the most significant SNP in the constant replicates maintain a frequency of about 75%, while the increasing replicates drop to approximately 25% by week 6 and then plateau. The 2 Roundup treatment allele frequencies move in opposite directions, while the control appears to have its own intermediate allelic signature. Although there is quite a bit of variation between the control replicates, the downward trend in many of the replicates suggests the control populations might be under weak directional selection. The constant is likely under stabilizing selection, while the replicates of the increasing treatment move in opposite direction, inferring potential directional selection with uniformity among the replicates. It is interesting to note that in the increasing treatment, as the Roundup dose increases at week 6, the allele frequency flattens. This potentially implies that the frequency may again increase if we were to apply a prolonged exposure of a stronger dose beyond the initial 10-week experiment. This region might be involved in Roundup dose-specific responses, implicating that this site may have pleiotropic consequences. Circling back to previous research, we did not observe significant allele frequency changes in a priori candidate genes mentioned previously, DIP5, PDR5, or AR1. We did, however, independently identify 2 interesting potential candidate genes, HAL9 and ENA1. HAL9 is a putative transcription factor that activates ENA1, which further activates several ABC multi-drug transporters, including PDR5. We also observed 24 genes that overlapped with the 2 previous RNA-seq and mutation accumulation experiments mentioned previously, 15 of which were identified within Roundup-specific peaks and 9 within constant-specific peaks. While we cannot say that these genes are involved in Roundup resistance for sure, based on experiments to date, they are strong candidates for further functional genomics investigations of Roundup resistance. There are 2 genes that overlap in all 3 experiments, NRG1 and BAP3, with their corresponding annotations listed on this slide. These 2 genes are 2 strong candidates for further functional investigation with regards to glyphosate exposure. It may be surprising to you that there's little overlap between the genes we discovered and the 1,000+ genes that were identified in previous studies. But there are several key distinctions in our methods that likely explain these differing results. First, the lab that conducted both of these experiments used a different commercial glyphosate formula that has a higher percentage of glyphosate and potentially different additive ingredients in Roundup. Secondly, different growing media conditions were implemented to identify some of these genes. And finally, these experiments were performed using a handful of individual isogenic strains that lack the diversity that our 12X ancestor obtains. So while there are many possible reasons why our experiments may have identified different candidate genes, we are not surprised to see additional putative genes that do not overlap due to the rich genetic background that we are working with. These genes are also strong candidates for further investigation, as they may highlight new genomic sites leading to Roundup resistance. With regards to our second research question of whether our increasing treatment has improved resolution to detect candidate traits, we would expect to see wider peaks with more genes being identified in the constant treatment, while the increasing treatment should have narrower peaks identifying fewer potential candidates. It was hypothesized by Velachos and Koffler in their 2019 paper that implementing an increasing selection treatment, which decreases the number of selected individuals over time, reduces such hitchhiking and leads to higher power to detect specific alleles underlying adaptive traits. However, our increasing treatment did not reliably enhance the resolution in which we could detect candidate regions. We did not observe a consistent pattern that aligned with this expectation, as the first 2 pairs of Roundup-specific peaks were narrower and identified fewer SNPs in the increasing treatment comparison to the control shown here, while the 2 other peaks pairs revealed the opposite pattern of having narrower peaks in the constant treatment. There are several possibilities for why we did not observe obviously narrower peaks in the increasing treatment. One reason might be the dose of Roundup. So even though our dose of Roundup steadily increased over the weeks of the experiment, this may not have led to associated increases in selection pressure each week, since over time we would expect the populations to become more resistant to Roundup with repeated exposure. A more pronounced weekly increase in the dose of Roundup might have alleviated this concern. Another important aspect to consider is the number of generations that occurred under Roundup selection. While our experiment featured over 100 asexual generations in the Roundup selection treatment, only 10 discrete cycles of sexual recombination occurred. The work by Velachos and Koffler simulated evolution using sexually reproducing fruit flies, which have different rates of recombinations. So we cannot directly equate our generation estimates in our yeast system with a single generation in flies. It is possible that with a longer experiment, we might have observed larger and more consistent differences between the two treatments. In summary, regarding our first question of the genetics of Roundup resistance, we did not observe a substantial difference in phenotype between the two Roundup treatments. However, we did identify significant allele frequency changes that vary between the treatments. We observed signals of significant genomic change in several a priori candidate genes and also discovered new putative candidates at other loci. We also found 4 shared peaks between the constant increasing regimes that are strong candidate regions for Roundup resistance, and observed 2 additional peaks that were independent to just the constant treatment, suggesting that the genetics of Roundup resistance is likely dependent on the strength of exposure. Concerning our second question, we observed different genomic signatures of adaptation between the 2 Roundup treatments. While there is no clear pattern of narrow peaks with fewer putative candidate genes being implicated in the increasing treatment, we are seeing more peaks that are specific to just the constant high selection pressure. Implementing 2 different selection treatments revealed a unique trait architecture of Roundup resistance that implies that different alleles are selected for under different Roundup doses. These findings highlight the importance of implementing different selection strategies when implementing experimental evolution experiments. This study identified several genomic regions that may contribute to improved Roundup resistance in yeast and further add to the body of work that is seeking to understand how non-target organisms evolve in response to chronic exposure to this herbicide. Next steps would include performing functional genomic assays of strong candidates to further verify their potential contribution to improve Roundup resistance, especially the genes that overlap with results from previous studies. If naturally occurring allelic variants in the genes can be definitively linked to a functional increase in resistance to Roundup, these variants could have value in an applied context. Herbicides like Roundup appear to affect natural microbial communities, particularly in soils whose health is critical for large-scale agriculture. By identifying alleles that allow microbes to thrive in the presence of this product and potentially also directly metabolize it, our basic research may inform applied bioengineering efforts to optimize soil microbial communities for increased crop yield and for bioremediation of soil toxins such as broad-spectrum herbicides. These efforts could lead to improved farming practices that increase yields and safety. I want to thank you all for coming and listening to my talk today, and I especially want to thank Dr. Mollingberg for her mentorship over the last 2 years. It's been a pleasure being a part of the Burke Lab, and I'm definitely going to miss everyone as I move forward and hope to stay in touch. I also want to thank Savannah Taggart. She's a Sure and Nurse Engaged Scholar here at OSU, and she is an undergrad who helped me greatly with my research project. And I just want to thank her for all her hard work and her perseverance despite the conflict or many challenges we had with COVID during my experiment. And then I just want to reach out and say thank you to my committee members for their support and their criticism of my work. It's made me a stronger scientist, and I look forward to engaging in science in the future. So thank you. And an additional thanks to my office mate, Mark. It's been a pleasure, and all the Burke Lab members. I'm very happy to have ended up in a lab with a lot of friends that have made this process a lot of fun, and hope to stay in contact with everyone. All right, I open it up to questions now.
Yeah, thanks very much, Chloe. I'm happy to step in and help with the questions to the extent that I can. So if anyone wants to ask a question, feel free to do so either in the chat or by raising your Zoom hand. And between me and Chloe, we should be able to see who's asking things. So it looks like Anne, you have a question. Go ahead and unmute yourself and ask.
Hi, Chloe. That was such
Hi.
an awesome presentation. Well done. I was wondering if you could explain to an ecologist a little bit more about what's the overall variation in Roundup resistance that you get from the ancestor yeast population. I guess I'm trying to get a sense of how do you obtain, like in the lab, variation in ancestral populations, and whether your choice of strain might affect your results.
Yeah. So due to the time limitations of my master's, I started with a population that already existed and had been in the lab for a while. I have, I think, a slide down here that kind of shows. So this is actually from Savannah Tagg's, the undergraduate researchers thesis, who helped me with the project. And these are the different strains within the 12X population in 1% Roundup. So we are seeing quite a bit of variation. This was actually done, the phenotyping was actually done after I started my experiment because I didn't have the luxury of time with a short master's project to do this work ahead of time. But we were able to see that there was quite a bit of variation within the population. So in an ideal world, I probably would have done a lot more for a PhD research. I would probably be looking at different populations and doing a little bit more investigative work about the effects of Roundup on them before implementing it. But because we had a bunch of different isolates from different locations and they were isolated from different environments, we thought that that would be the most advantageous strain or population to use. More phenotyping work kind of showed here, these two columns are the most informative here, but the doubling time rate of these populations varies widely. And surprisingly, we didn't necessarily see agricultural strains here shown in orange, or sorry, fermentation strains shown in orange. Didn't have a consistent pattern that they had a improved growth rate, because we see some down here that have really long doubling times. So I think in some ways, some of these expectations that we maybe thought that these strains might have had previous exposure to chemicals and might have helped them weren't necessarily true, but it seems like we just need, if in continuing this research, we would need to do a lot more work to kind of untangle why that's the case and how the phenotypes are being affected. Thank you, Anne, for your question.
All right, I see Stephanie with a question. Go ahead, Stephanie.
Hi, first of all, excellent job, Chloe. I really enjoyed hearing all about your research. I was just curious, you mentioned with the control group and how you were having to, I think, some of the work, bringing them through each week, there were maybe some other chemicals within the Roundup that were maybe similar to some of the other chemicals you were using. I didn't know if you were interested at all at differences between the main component of Roundup versus some of the other chemicals, or were you just focusing on everything all together just to see what its effect was?
Yeah, so for this experiment, I was kind of limited, especially since we were doing the constant and increasing treatments. So I wasn't able to include glyphosate as a control, but I think it would be really interesting for a larger project to investigate glyphosate next to the full commercial formula. There's a couple other, for the most part, the ingredients are undisclosed and they change. So it's hard to test exactly what those chemicals are without having the inside knowledge of what they are. But that would be definitely something that would be interesting to me to investigate in the future. But yeah, it's good to hear your voice, Stephanie. Thanks for showing up.
Sure. Good to see you. Thanks.
All right, looks like Mark has a question.
Yep. Hi, Chloe. It's a great talk.
Hi, Mark.
Yeah. So I just had a question because I noticed the example SNPs you showed, so like the most significant ones in each peak, those were all at fairly high starting frequencies. Was that kind of characteristic of all of the SNPs in those peaks, or was that just like a quirk of the one that was selected as the most significant?
So in the plots, I also phased those so they would all be in the same direction. So not all were at the same starting frequency, but for the ease of explanation, I kept them that way. So some were lower, some were higher, but in general, the trends within the plot were the same that we were seeing. I have a few more trajectories here that kind of demonstrate the patterns, but these are, keep in mind, they are phased. We see kind of more variation within the replicates within these 4 plots that are Roundup specific, and these are all the top SNPs from those peaks. But we see kind of in a lot of them a kind of flat selection, stabilizing selection, what appears to be or what seems to be the case, while there's kind of a decrease here in this one. So there is some variation more variation within the replicates of the other top SNPs.
Okay, yeah, no, thank you. I was just kind of curious if there was just an enrichment for things that were just at really high near fixation and then they stay there or something like that.
Yeah, and it wasn't always the case. There were some with lower frequencies within the constant increasing treatments.
Okay.
And they stayed.
All right, well, I don't see any more Zoom hands or any more chats, although I'll talk slowly just in case someone wants to add anything at the last second. But if no one else has any questions, then I think our next step is to convene as a committee. So the committee has a separate link to join in an email, so we can conclude the public forum of the thesis. Unless Teresa, please chime in if there's anything I'm forgetting to do that I should do.
Nope, good.
Yeah, but if not, then let's just thank Chloe for her talk with your virtual applause and say goodbye for now.
Yay.
Thank you, everyone.